Hello to all the learners and viewers of Gyan Darshan. Myself, Dr. Ravi Rajwanshi from Discipline of Life Sciences, School of Sciences, IGNOU. Today, I'm going to talk about a very interesting topic, in fact, a subject called biotechnology. The title of my today's lecture will be Introduction to Biotechnology. This is from the program of BSc Journal of the course Economic Botany and Biotechnology, bearing the code BBYET143. So to start with, this term biotechnology sounds really interesting. So do you know what is biotechnology? The answer is hidden in the term itself. If we divide this term into two parts, one is called bio, the other one is called technology. So bio means life. So you are using some life forms in connection with or with the utilization of technology for the benefit of humans or mankind. So this is a short definition. Let's start with the detailed definition of the same term, biotechnology. So this term, biotechnology, was actually coined by a Hungarian engineer named Karl Erecke in 1919. So at that time, this term include all the processes by which products are obtained from raw material with the aid of living organisms. So in the slide you can see the picture of Karl Erecke. Later, this term was defined by Convention of Biological Diversity, in short CBD, where they defined biotechnology as any technological application that utilizes biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. So this term was given in 1992. So this definition is applicable to both traditional and modern biotechnology. So we'll be coming in details that what actually is traditional and modern biotechnology. So this question comes into our mind, that is this technology or this science, this branch of science, is really a very new one, or it was existing since long? So let's discuss the same first. So there were various stages in the development of biotechnology. If we have to divide these stages, we can divide it into three types. The first one is ancient biotechnology, the second is classical biotechnology, and the third one is modern biotechnology. So coming to ancient biotechnology, the period for this can be given between the 8000 to 4000 BC. So just remember the definition of biotechnology I have given just now and try to correlate if these examples are in agreement of the definition or not. So the biotechnology actually existed since long time. In the early history, the biotechnology can be related to the domestication of animals to get milk or meat. So you know that humans have started feeding on animals and getting the food from them in the starting. Later on, they start cultivating the plants such as rice, barley, and wheat as a main source of food, which can categorized as biotechnological development. So you can say the domestication of wheat, which is compared to ancient biotechnology, the domestication of the wheat can also be considered where it has been the genes have been manipulated, or it has been changed in such a way so that the productivity is enhanced and less damage to the crop can occur. So cultivation of plants is the second step which we can correlate with biotechnology. After cultivation, the next step was learned by the humans was to breed. With the observation, they started learning that by the process of breeding, they can generate or develop a progeny with desirable characters. So you can see that breeding was also not new. It is based upon the observation and selecting the right progeny for further propagation. So you can see here that this, starting from cultivation to breeding, can be given under the broad umbrella of biotechnology, or you can say the ancient biotechnology. After ancient biotechnology, which was long back, then comes the era of classical biotechnology. Now, the classical biotechnology, you can say it was during the period of 2000 BC or 1800 to 1900 AD. The age-old practice of production of cheese, yogurt, bread. So you know that these things are not new. These have been made since long time. But at that time, the humans were not aware that is there any microorganisms involved behind the production of these products. With the invention of microscope, we got the flexibility to visualize the microbes we were not able to see them by the naked eyes. So that flexibility we got after the invention of microscope. And we started learning about the microbes, microorganisms, and then characterizing them. And after that, we are in a situation, now we know exactly what are those microorganisms which are basically involved behind the production of these products. For example, for cheese, it's lactococci, lactobacilli, or streptococci families. For yogurt, you can make it by lactobacillus or streptococcus thermophilus. Now bread can also be made by using yeast. Which yeast? Saccharomyces cerevisiae. So you can see that these are one of the well-known examples of classical biotechnology we can always quote. Apart from this, manufacturing of wine, alcoholic drinks, is also not new. Since many ages, these drinks were made by for human consumption, but the process behind this was not given any term. So manufacturing of alcoholic drinks like beer and wine via fermentation, I'm using this term fermentation here because now we know that this process is called fermentation, but earlier it was not known that what is actually it is. So by using the process of fermentation, we can make these alcoholic drinks. In fact, role of microorganisms such as bacteria, yeast, and molds in hydrolysis of sugars for the production of ethanol was also observed by humans, and hence this technique of fermentation was discovered. Later in 1796, Edward Jenner demonstrated the role of vaccination in providing immunity against smallpox. Hence, he is also considered as the founder of vaccinology. Dear learners, you must be very much aware that very recently the world has faced a pandemic in the name of corona virus, and the life-saving strategy was either to develop vaccine or to develop herd immunity for protection of human beings from this deadly disease. So you can see presently in 2024, the development of vaccine has a backbone right in 1796. The principles remain same, but the technology advances. Maybe in my future lectures, I will be discussing these things in much detail, but this is the introductory part, so I will be quickly going through the major introduction part only. Now, scientific evidence for fermentation was described by Louis Pasteur in the late 1800s. He proposed the theory as germ theory of disease. Just imagine at that time, it was a very important hypothesis given by Louis Pasteur, which became the base of modern medical biotechnology, where he said that there is a role of certain microorganisms known as pathogens or germs that can cause disease. So, in fact, the product like honey, soya bean curds, which are the natural products, surely, but these can also be correlated with biotechnology? Yes, the answer is yes. It can be correlated with biotechnology when you are using the honey, which is a product or saliva of bees, honeybees, to use it as a natural antibiotic. And it is also used for healing the wounds since ancient times. So you can also see that these examples also satisfy the condition laid down for the definition of biotechnology. Even Ukrainian farmers, they used moldy cheese to treat infected wounds. You have to see that development of biotechnology actually requires a very keen observation. How they came to know or how the antibiotics were actually developed, it was observed that during a random infection, that bacterial infection around the mold, it was found that there was a zone of inhibition. Means the bacteria was not able to invade the mold at certain diameter around the mold. So, scientist or the person who has observed, he thought that there must be something there in the mold which is not allowing the bacteria to invade it or to infect it. And hence, this gave the foundation for the identification of antibiotics. And now we are using so many types of antibiotics nowadays to cure many types of diseases. So these were some of the examples from ancient biotechnology. But now we are coming to modern biotechnology. Now the question is, what is the difference of modern biotechnology from the ancient or classical biotechnology? So by the time of 1900 to 1953, the subject of genetics has also started to develop. And DNA research was also initiated between 1953 to 1976. So in this era, the main emphasis is on the biological molecules, that is DNA, RNA, protein. So these biomolecules are the main target for developing the new products. And for this, the genetic and using this information, it is now possible to transfer the genetic information from one organism to another organism. How it can be transferred, we will be discussing in the coming slides. The production of insulin and the first restriction enzyme was also discovered in 1970. This discovery basically enabled the researchers to insert the foreign gene into a microorganisms, which was initially bacteria. Now it can be done in other microorganisms too, but it was starting with bacteria. And this discovery of restriction endonucleases laid down the foundation of recombinant DNA technology, and this is, you can say, considered as a birth of modern biotechnology. In 1977, in Scotland, a sheep named Dolly, this is a well-known example, many of you must be knowing this name. It was cloned using the adult somatic cell. And later on, another sheep named Polly was also cloned from the somatic cell, but this was having an additional gene and hence called transgenic animal, and this gene was transferred using nuclear transfer technology, as it had the gene encoding for human clotting factor 9, which is a protein involved in preventing hemophilia. So, coming to modern biotechnology and with special emphasis on recombinant DNA technology, let me explain you what is this recombinant DNA technology. It is the process of constructing and manipulating DNA sequences that do not occur naturally by combining DNA fragments from different sources. So, as I said in the previous slide, that recombinant DNA technology gives you the flexibility to transfer the gene from unrelated organisms, from one organism to another organism, and hence the barrier of sexual incompatibility has been overcome because of this technology. So, to start with recombinant DNA technology, there are certain few steps that you have to go through. The first is isolation of gene coding for a protein of interest, then cloning or the transfer of this gene into an appropriate host cell, then improving the gene or protein expression by using a strong promoter. So, during the cloning process, you can not only clone a gene of interest which code for a protein of interest, but you can also regulate its expression level using an appropriate promoter. So there are different types of promoter. For example, there are constitutive promoter which can constitutively express the gene of interest, whereas there could be inducible promoter which can express a gene at a certain condition when a proper trigger is given to it. That trigger could be a chemical compound. If you are using a stress inducible promoter, then you have to apply the stress, and under that condition only that gene will express. So this all things, transfer of gene and other regulatory elements, basically resulted in the development of genetically modified organisms. So the question is, what are GMO or genetically modified organisms? These are the organisms whose genetic material has been modified, or for example by introducing foreign DNA sequences obtained by recombinant DNA technology, and hence the production of transgenic animals and plants was only possible because of recombinant DNA technology. So in short, I can say that any organism which is having a foreign gene is called as a genetically modified organism. So if it is a plant, then it is a genetically modified plant. If it is an animal, genetically modified animal. Or if it is a food product, that's why from a genetically modified organism, then it is also called as GM food. And these things has to be properly labeled. So biotechnology do not just provide a solution to many problems, but at the same time, it is also self-regulated due to many biosafety laws and guidelines. So these guidelines are very important to be followed so that there should not be any escape of any genetically modified organisms prior to its proper permission from the competent authority. So, if I have to summarize the overview of the recombinant DNA technology and what were the major inventions or discoveries which has actually laid the foundation of this technology, it should start with the description of double helix structure of DNA by Watson and Crick in 1953, then followed by development of genetic engineering technique by Cohen and Boyer in 1973. Then first human protein, somatostatin, was produced in bacterium called E. coli in 1977. The first commercial use of protein, that is human insulin, was given permission in 1982. A very important technique of polymerase chain reaction, in short we called as PCR technique, was also developed in 1983. The first genome sequence of an organism, Haemophilus influenza, was determined in 1953, and a very important project called Human Genome Project, that refers to international 13 years of efforts, was started in October 1990 and was completed in 2003 with a discovery of approximately 20,000 to 25,000 human genes. And these genes are now, the sequence of these genes are now available for further research. For this, there are different types of gene banks where the sequences are submitted, and these sequences, there are so many sequences of prokaryotes and eukaryotes, now they are available in gene bank. So this particular aspect is basically covered under the bioinformatics subject. Now, let's see that what are the different types of biotechnology. Biotechnology can be of different types, and that types are denoted by different colors. So these subdisciplines can be divided into red biotechnology, that involves development of new drugs, vaccines and antibiotics, regenerative therapies, molecular diagnostics, and genetic engineering techniques to cure diseases applying genetic manipulation. The other is white or gray biotechnology, which includes designing of energy efficient, less polluting and low resource consuming process and products. After this comes green biotechnology. From the color itself, you can correlate. It covers the agricultural processes such as producing pest and disease resistant crops, animals, and environmentally friendly agricultural practices. Followed by this, there is blue biotechnology. From the word blue, you can also again correlate that it involves use of marine resources to create products and their application in various sectors such as converting aquatic biomass into fuels and pharmaceuticals. Then yellow biotechnology, that refers to the use of biotechnology in food production under such as making wine, cheese and beer by fermentation, that I have already discussed. Then comes gold biotechnology, that, as I told you, this basically deals with the bioinformatics aspects, which utilizes computational techniques for the analysis of biological data. Then comes brown biotechnology, which is also related to green biotechnology, and it aims to boost the richness of flora of arid and desert areas by incorporating high resistant plant species into the soil. After this comes the violet biotechnology, that ensures the practice of biotechnology is in compliance with the law, ethical standards governing each field and philosophical issues related to biotechnology. So I would say, if these ethical issues are properly addressed, then there is no scope of the next biotechnology, which is termed as dark biotechnology. And this dark biotechnology basically is defined for using the biotechnology for bioterrorism, making biological weapons and biowarfares using microorganisms and toxins that can cause diseases, death in humans, domestic animals, and crops. So, we'll be discussing some other aspects after a short break. So, we'll be joining back. Keep tuned.
The process responsible for the whole manufacturing of our clothing. If you want to know more about this, Indira Gandhi National Open University from the School of Continuing Education has launched a program which is Certificate in Apparel Merchandising. There are many benefits after doing this program. You can become a designer in various fields. It can be fashion designer, apparel designer, costume designer, or even a textile designer. One can also work as a fashion consultant.
Thank you.
Research and development is another field which one can take up after doing this certificate course. Going into academics is another perk after doing this program. Getting into fashion houses or fashion studios as an intern is also a very good option post doing the certificate in apparel merchandising. The basic eligibility of the program is 10+2. The duration of the program is 6 months to 2 years. The program is also available for both the admission cycles, which is July and January. The exams are conducted twice a year, in June and December. So what are you waiting for? Join this program and get an enriching knowledge in the field of apparel merchandising.
ठीक है हाँ ठीक है Welcome back, viewers and learners. Before this short break, we have discussed some definitions of biotechnology and some history of biotechnology, their stages of development. Now, we are going to discuss some applications of biotechnology in different fields. So, broadly, you can say that biotechnology can be applied to different fields, you can say subfields of biotechnology, which can be classified as plant biotechnology, animal biotechnology, agricultural biotechnology, environmental biotechnology, industrial biotechnology, and aquatic biotechnology. So, coming to plant biotechnology, which is the main focus area of this particular course on botany, you can use these biotechnology tools for developing different types of abiotic stress tolerant plants. As discussed earlier, recombinant DNA technology in addition to molecular assisted breeding can be employed to develop the abiotic stress tolerant plants. So what is meant by abiotic stress? First of all, So there are any physical factors which can hamper the growth of a plant, maybe salt, drought, heat, cold, etc., can be considered as the abiotic factor. So you can develop the crop plants which are resistant to these extreme abiotic factors. Next, you can, in the similar way, you can also develop the transgenic plants for biotic stress tolerance, where the biotic means any type of stress which is caused by any type of living organisms. For example, it can be a viral disease, it could be a bacterial disease or a fungal disease in a plant that can hamper the growth and productivity. In addition to these, insects, herbicides can also affect the productivity of a plant or a crop. So these all things are covered under biotic factors, and this biotechnology give you the flexibility to develop the plants for these types of tolerance. Then, as discussed earlier, this can be applied for value addition. For example, you can make a genetically modified crop which are widely used, for example, in United States of America, which is quite popular, these soya beans, corn, sugar beets, canola, cotton, all these things are commercially grown, and some of them are used as GM food cash crops. Similarly, cereal crops like rice, wheat, maize, oil seeds like soybean, lupin, sorghum, tuber like potato, sweet potato, cassava, beet, fruits and vegetable like tomato, and many other plants have been developed as a source of GM food. So you must have heard about flavor saver tomato. Now, there can be another application of plant biotechnology, that is in pharmaceuticals. You will be surprised to know that plants can also be utilized to produce edible vaccines. As we know that vaccines were earlier produced through microbes only, but this plant biotechnology basically provides you the tools where you can clone the genes for certain antigens against which the immune response can be generated into the plants. So edible vaccine concept came into existence. So genetically modified plants can produce encoded proteins. They are bio-friendly, they are also cost-effective and easily administrable. It is easy to store and it has wide acceptable options, because if you can consume a vaccine in an edible form will be a more acceptable option compared to if you are having needles to inject them into your body, which is quite painful. So usually these type of vaccines are developed in the target plants which are consumed raw. Maybe banana, tomato, carrot, corn, peanut, potato. These are some of the plants or crops that can be targeted for developing edible vaccines. Whereas tobacco is considered to be a model plant. Before applying any technology into a crop plant, it is initially tested in a model plant, and tobacco is one of them. So production of recombinant therapeutics is another option which is possible due to biotechnology, or I should say plant biotechnology, to the present world. Anything that is commercialized can come under the broad heading of industrial biotechnology. So there are certain industrial products, for example, plants that produce small number of enzymes such as avidin or beta glucuronidase. In short, it is called GUS. This GUS gene is also utilized as a visible marker while developing transgenic plants. So we are going to discuss that where it can be used in the coming slides. Then Bioplastics is also one of the option which can be developed using plant biotechnology. Then what is bioplastics? Bioplastics are the substances that are made from organic biomass sources where polyhydroxyalkanoate is the major component for its production. Apart from this, production of biofuel can also be achieved using transgenic plant. So biofuels can also be, like Jatropha is one of the well-known example where such biofuels can be produced. Although this particular unit is mainly focused on plant, but let's discuss few examples from animal biotechnology also. So biotechnology have a role to play in stem cell research. What are stem cells? Stem cells are the specific type of cells which are capable of evolving into many different types of specialized cells. Means these are types of cells which can be programmed to develop into a particular type of cell. So it has a very important scope to treat many types of diseases, which, for example, specifically through a technology called stem cell therapy. So the scope of their treatment using the stem cells can be in the diseases like diabetes, cancer, Parkinson's disease, Alzheimer's disease, even wound healing, male infertility. So it has many applications, stem cell research. So animal cloning also, in animal cloning, drug testing trials also, these stem cells can play a very important role because science is a continuous process and it keeps on evolving. So these stem cells can also be developed into a cancer cell line where you can test different types of drugs on them and see how they are actually working before going for any type of animal or human trials. So productive cloning of somatic cells, nuclear transfer, SCNT, I have already discussed in the previous slides, is one of the option, like for the cloning of Dolly was one of the example. Then therapeutic cloning can be achieved by animal biotechnology where cells can be removed, are pluripotent and can give rise to all cells with an exception to embryo. So cloning can reduce the wait time for organ transplant because we know that presently we have to wait for some donor. So if it is possible to generate the organs by using these types of cells, it would reduce the waiting time as well as it will also put less pressure on any donor because if someone is donating an organ, surely it has to compromise with its functioning of the body part. So there are many immunological concerns which can also be addressed if the organs can be developed using this technology, because during the organ transplant, the graft rejection or those possibilities can also be ruled out if we can use this type of technology. Then the role of animal biotechnology can also be seen in biofarming. So GM plants and animals that can produce substances with pharmaceutical importance can be developed. So animal biotechnology have also a role to develop transgenic animals, and many such animals have been developed for research purpose like mice, pigs, rabbits, rats, fishes, sheep that has been created using the transgenic approach. They can be useful for scientific research, improving the traits, and for example, you can also improve the milk quality. You can develop a breed which is resistant to disease. That can be animal, that can be poultry, that can be fish. So production of donor organs, as I have already discussed. So these can be achieved using animal biotechnology. Insulin-like growth factors, IGF-1, then human nerve growth factors, HNGF, human growth hormone, recombinant human serum albumin, etc. There are many examples which can be produced now by developing a transgenic animal. So, another application lies in the transplantation and drug development, as discussed, cell therapy, tissue engineering, generation of organs from simple adults, tissues, stem cells, or by using a blastocysts complementation system. This technology can be helpful. Therapeutics, diagnostics, and molecular medicines. Animal biotechnology has a role to play in this also. Here, you can produce human insulin, you can produce monoclonal antibodies, human growth hormone can be developed, can be generated, vaccines can be generated, and gene therapy can be done by using animal biotechnology. The others are breeding. As we know that artificial insemination, in vitro fertilization are also today's need when we have to artificially multiply some animals, or even in humans, these technologies are now, IVF is widely used for ruling out any problems for infertility. Somatic cell nuclear transfer is another technique that is widely used in animal biotechnology. So coming to agriculture biotechnology, I would say this agriculture consists of what I have discussed in animals as well as in plants. There are both the things are covered under this broad umbrella of agriculture. Agriculture not only just deal with plants, it also deals with animals also. But our prime focus here in this particular unit is on the plants. So you can quote all those examples of plants in agricultural biotechnology, where it can be subclassified into crop biotechnology, horticultural biotechnology, tree biotechnology, food processing, or plant biotechnology. So there are, coming to the specific topic of development and application of transgenic plants, that has already been achieved and many things are still in progress. I'm going to just summarize how these transgenic plants can be helpful in agricultural biotechnology. You can develop the plants for nutritional value, like for seed storage proteins, amino acid composition, carotenoids, vitamin E, plant lipids for food use. You can also use or develop transgenic plants for agronomic traits, such as for inhibiting seed dormancy, for developing herbicide resistance, insect resistance, disease resistance against different microbes like bacteria, fungal, or virus, nematode resistance. Then also you can use to develop, as edible vaccine example I have explained in the previous slides, to produce recombinant proteins. You can also develop plants for phytoremediation. What is phytoremediation? Phytoremediation means the plants which can be utilized for removal of any type of pollutant which is present in soil, or if it is a water plant, in water. So for that purpose also, pharmaceutical compounds, as discussed in the previous slides, can also be produced. Horticultural traits, for example, novel flower color. You see different types of flowers in the market. So by using these approaches, you can develop different types, textures, colors of flowers, so which might have a more better commercial value. Then food and industrial traits can also be influenced, like for wood fibers, fruit ripening, means you can delay the fruit ripening. If suppose any fruit has to be transported from one continent to another, it would be better if it can be harvested at right time, and during the course of transportation it should not get ripened by the time it reaches market. So you can use this technology for that. Then plant productivity, as discussed earlier, like stress tolerance, can be achieved, both biotic and abiotic. Metabolic pathway can be regulated. For example, nutrient capture from soil, carbohydrate enhancement, or formation of novel oils, essential oils can be achieved by this transgenic approach. Coming to environmental biotechnology, as the term itself says, the biotechnology which applies its role in environmental monitoring, waste management, pollution prevention. So these all can be covered under environmental biotechnology. The best known examples are the development of genetically engineered microorganisms. In short, we can call it as GEMs. So you must be knowing about a very well-known example of superbug, which was developed by Anand Chakrabarty et al. In 1979. This superbug was a constructed bacterium, Pseudomonas putida, that can degrade hydrocarbons found in petroleum wastes. So it was a multigenic engineering approach. So you can clone multiple genes also. That is called gene pyramiding, to finally get rid of pollutants present in such places. Then comes industrial biotechnology. So any commercial use of the products come under this category. If I quickly go through it, it is a metabolic production, for example, acetone, butanol, alcohols, etc. Then methane production by anaerobic digestion, waste management treatment, waste treatment, production of biocontrol agents, fermentation of food products, bio-based food and energy, recovery of metals and minerals, production of bioethanols. All these things, even pharmaceuticals, everything has a commercial value. So everything can be, these examples can be covered under industrial biotechnology. Then comes your aquatic biotechnology. From the term itself, you can make out that it is the role of biotechnology in aquaculture, restoring and protecting marine ecosystems, improving seed food quality, environmental remediations, marine byproducts for human health, biomaterial and bioprocessing, marine molecular biotechnology. So, coming back to the same topic of our plant biotechnology, there are different methods which are the main methods of plant biotechnology. The first one is tissue culture, followed by second, which is called genetic engineering or recombinant DNA technology. Then comes molecular assisted breeding. In this slide, you can see that what is plant tissue culture, because for developing any type of transgenic plants, maybe the first step could be the plant tissue culture. The plant tissue culture has a very important role to play in biotechnology itself. Plant tissue culture is a technology or a technique to grow cells, tissues, organs, etc. in controlled and aseptic conditions. Plant cells are totipotent and hence they can give rise to an entire new plant. So there are different applications of plant tissue cultures. These applications can be in clonal multiplication, conservation of germplasm, raising of virus-free plants, embryo rescue, production of haploids, soma clonal variations. So even if the plant is, for example, generation of virus-free plant is possible, if there is a germplasm which is completely infected by a virus, the cells you can take from the apical meristem, which multiplies much faster, and they do not have much developed plasmodesmata, so that the viruses can easily cross during to spread their infections. You can just take out those cells and multiply them into a complete new plant which will be free from virus. So we can discuss these examples in my future lectures. Here comes an overview of plant genetic engineering. Here in the picture you can see in the inset there is a bacteria which is called as natural genetic engineer. If I ask you what's the name, that is the Agrobacterium tumefaciens. Under natural conditions, this particular bacteria have 3 genes present in its tDNA which code for auxin, cytokinin and opin. These are responsible for causing crown gall tumor in the stem area, as you can see in this picture. This is a crown gall tumor above the soil in the stem. So scientists has exploited this particular phenomena for self-benefit. In this technique, they have disarmed these genes with the gene of interest, and then they are cloned into suitable host. After cloning, they are transfected. These clones, like agrobacterium, is infecting the callus or suitable explant. After infection, they are grown on MS media or any suitable media having selection marker, maybe some type of antibiotic, and those which can successfully grow are considered to be putative transgenics, and further they are multiplied for further analysis. Here you can see the picture, which is from my own research article. Here you can see the pictures which can clearly show that this A is the callus formation, where you can see a small shoots are coming up. Then these shoots are growing, and you can see some leaves are coming up. Followed by this, these shoots are cut and they are put into rooting media. Once the roots are there, they are properly, carefully taken out, as they are equivalent to a small baby, which were very delicate. So they are taken out and they are acclimatized to the outer conditions. So they are planted in an agro pit. And once they are hardened, they are transferred to the greenhouse. So in this picture, you can see the center one is the control plant, which is not a transgenic or untransformed control, whereas the other two are the transgenic plants. So you can see there is no difference in the morphology or the productivity of the plant when we develop them in by transgenic approach. So there are different types of integrations. Like these integrations can be stable transformation, means your gene of interest has been stably transformed into the genome or not. So there are different types of integrations of the transgene into the genome. So you can quickly just see there could be a stable transformation, there could be a transient expression, there could be an integrative transformation. When the gene is integrated into the nucleus, it is considered to be nuclear transformation. If it goes into mitochondria or chloroplast, it is considered to be organelle transformation or episomal transformation. Next comes your marker-assisted breeding. As I told you that apart from rDNA technology, marker-assisted breeding is also another technique that can be used where some types of indirect markers are seen or observed, which are cloned, or you can say which are present, not cloned, if they are present next or near to the gene of interest. So if you can screen those markers, you can also assume that your gene of interest is also present in the progeny. For that, different types of markers have been used. It could be morphological, biochemical, or DNA based. For DNA based, which are widely used, they are SSRs markers, RFLP, SNPs, AFLPs, and you can use a technology like PCR also, where you can directly look the presence of gene of interest itself. So this is a flow diagram which shows the technique of marker-assisted breeding, that how marker-assisted plants, using the marker-assisted breeding, the plants are screened, and finally the progeny is achieved that has the required traits which are beneficial for the humans. So to conclude, in this lecture, I have discussed about the biotechnology, the definition, their different time periods, how this biotechnology basically developed in a course of time, that is ancient, modern biotechnology, then the role of plant biotechnology. So to conclude, we can basically use this technology, means you can have every sort of solution available through this field of biotechnology. The only thing is you have to pursue your career, and I hope this lecture will be helpful to you and you will be going for future studies in the field of biotechnology and research too for the benefit of the mankind. So thank you all of you. So I'll be taking the leave from you people. Thank you.
Welcome and warm greetings,